Hello, my name is Alexis Caputo. Today's date is November 17th, 2018, and today I will be discussing biochemical testing for microbial identification for Bio 225, lab week number 4. For this lab, I will first discuss the second experiment, as the first part of the experiment requires multiple days for incubation. So the second part of the experiment is catalase testing of aerobic respiration, in which the test was conducted on Escherichia coli and Staphylococcus epidermidis, in which samples were collected from already incubated cultures. So these two right here, in which inoculation loop was to collect a small sample from each and placed within the black circles. And then from there, a drop of hydrogen peroxide was dropped on each culture, in which the test proved successful in that these are catalase positive microbes, as oxygen bubbles were formed when the hydrogen peroxide interacted with each culture. So the first part of exercise 1 is to transfer the media of the cultured bacteria into the MRV broth in order to incubate for over a course of multiple days. And then the second portion of the experiment after the incubation time is the methyl red test and bogus press scour. After performing the methyl red test, the E. coli kind of turns out like an orange color, which is a negative result, whereas the S. epidermidis turns out like a fuchsia pink, which is the positive test result. The negative test result could be due to after incubating for 72 hours, the E. coli never really appears turbidity. It's still translucent, so I'm proposing that's why the test is negative. After sitting for 30 minutes, the bogus press scour test results for both samples came out negative. Again, I know for sure the Escherichia coli test results are negative because the starting sample never showed turbidity, and I'm not too sure why the Staphylococcus epidermidis results are negative. So in conclusion, the Staphylococcus epidermidis produced negative results for the presence of butanediol, but positive results for the mixed acid fermentation of the methyl red test, whereas the Escherichia coli was negative for both tests